Okay, so welcome everybody to the next presentation. It will be about combination of quantum sensing and life sciences. Maybe not the very common combination. It's a new application in sensing and metrology. My name is Fedor Zeleska. I am representing Center for Integrated Quantum Science and Technology, Center for Quantum Biosciences at Ulm University. And here are the variety of applications that are coming basically into play very recently. So qubits as sensors for environment inside of cells, that will be the main topic, and we can apply it to a variety of parameters, starting from pH to nuclear magnetic resonance signal, temperature sensing. This is all very exciting, but also very unusual. So it's not the mainstream application of quantum sensors yet. But on the other hand, physics as a field was very successful in developing new tools for life sciences and medicine, starting from optical microscopy that gave us understanding of structure of cells, to modern reconstruction techniques like X-ray spectroscopy or electron microscopy. These techniques, developed by physics, gave us really a very crucial, very essential understanding of life. And it might be a moment now to apply quantum technologies for understanding of light at with a new, deep level. And today I would like to focus on nanoscale and sensing in cells, and techniques that are available, like for example electron microscopy, are of course capable to see individual molecules, but they usually deal with them under high vacuum condition. And if you ask biologists about a dream microscope, what could be a system that they would like to have at hand, they would say that we would like to see a single molecule without averaging. We would like to image individual atoms and molecules to understand the structure. We would like to see dynamics, the motion of molecules on a microsecond scale, because dynamics is linked to a function. But this is not all. It should be also possible to do in live cells under conditions where these molecules are acting, having a function. And there is no technique so far available to do this. Not in quantum sensing, not in classical sensing. There is no microscope which can fulfill all these requirements. But some techniques are coming quite close. And I would like to highlight two here, which will be relevant for this quantum sensing view on this field, namely optical microscopy and magnetic resonance. Optical microscopy, especially fluorescence microscopy, is a golden standard now in biology, in medicine. It is very sensitive technique because we can collect visible photons and detect them very efficiently. It is possible to image or track individual fluorescent molecules inside of living cells. So sensitivity is reached at the highest possible level. Spatial resolution, especially with the new nonlinear microscopy techniques like STAT, can reach about 10 nanometers. It's fast technique. It can operate with a video rate, but usually it is applicable for imaging labels, fluorescent labels. You don't see molecule itself. Many of them are not fluorescing or fluorescing very weakly, but you rather target your biomolecules by attaching a fluorescent labels. And this is technique which is very powerful, but not so universal, because you do image labels. So you cannot see inside of biomolecule. You hardly see the dynamics, except if you apply very special tools. Another approach, which is not a direct imaging, but rather spectroscopy, is nuclear magnetic resonance. In NMR, you are not focusing radio frequencies on the molecule. The wavelength is very large, it's meters. But in this case, you do a spectrum of the molecule. And because you have a very rich spectrum in nuclear magnetic resonance originating from the interaction between atoms comprising the molecule, understanding of interaction allows you to reconstruct this three-dimensional structure. This is very powerful technique. Contrary to electron microscopy, it can operate in liquids, but usually it requires many copies of the same molecule. It's not very sensitive technique. And if biomolecules are different, or if they are acting not synchronously, you are failing to capture dynamics, or at least dynamics in all the aspects. And you might also lose information about biomolecules if they are heterogeneous, because you will average your systems. So the combination of these two, reaching a single molecule sensitivity level, but also having a label-free understanding of dynamics provided by nuclear magnetic resonance is a very important goal. And so this is basically a field where many technologies are basically targeting to improve the sensitivity. Sensitivity in nuclear magnetic resonance is limited. You need these millions of copies because of two factors. One is very low polarization of nuclear spins and the ambient condition. Usually only one nuclear spin out of million really contributes to the signal, because you start with the thermal state. And there is a development in this area to hyperpolarize nuclear spin. There was our trade award in the area of quantum sensing given to company Envision, which is targeting to increase the signal in nuclear magnetic resonance by hyperpolarization. But today I'm going to talk about a different aspect, about improving sensors. Even though your signal is quite weak, but with a better sensor, you can capture the molecules with better temporal resolution and better sensitivity. And so I'm going to discuss application of quantum sensor to replace a coil in nuclear magnetic resonance spectrometer. So the sensor itself will be a quantum system. Quantum systems are very good sensors. If we are talking about nuclear magnetic resonance, we need a sensor for magnetic field to be very sensitive or very robust or very non-invasive. And in our case, we use a spin qubit in diamond. I will talk a bit more about this qubit. We approach it very close to atomic spin, and because if you are very close, you can be very sensitive. The fields are not very tiny if you are within a few nanometers distance, and you can be also very selective. So that basically will give you information about precession frequency of this nuclear spin. So we use a qubit as a mediator and measure the qubit basically approaching close to a molecule of interest, biomolecule of interest. The biomolecule itself is a classical object, so there is no really kind of insight now. I'm going to talk about quantum processes in biomolecules, but we are using tools from quantum technology, a quantum sensor, to make better measurements of the classical fields associated with biomolecules. The qubit itself that we are going to use is color center in diamond. When present in a high concentration in diamond lattice, it gives a coloration. This big color of the diamond is coming from these color centers as it dopants in diamonds that can absorb and emit the light. What is particular about this color center, why it is qubit, is because it carries a spin, a non-zero spin in a ground state, and the spin transitions, which are very well protected from environment and can be used as a qubit, as a sensor, are coupled to optical transition. By exciting color center with a laser and passing through the excited state, there are two processes happening. First, the intensity of emission of the color center is dependent on the spin state. So starting from one of these sublevels, giving you more fluorescence than starting from the other. And the second aspect is that elimination of the system lead to very high degree of spin polarization, even under ambient condition, which is relevant for biosensing. This spin can be prepared in a very pure state. So the spin temperature can be very, very low, although your diamond or nanodiamond is under ambient condition. And so those are two very essential tools. The third one is the ability to create these color centers on demand in a very pure diamond. And here engineering of diamond is very essential. Material science is very essential. So by having very pure diamond without any impurities, you can basically now start to implant individual atoms. And this array here represent a set of implanted nano-engineered color centers, and we can do this implantation with a precision of a few nanometers. And so for technology of sensing, where you want to approach your sensor to the biomolecule of interest, it is a very essential tool. Now the experiment itself, the sensing experiment, is a combination of these two techniques we discussed before. Sensitivity comes from microscopy, optical microscopy, where you can detect impurities with very high efficiency. So we use an optical microscope with very sensitive for visible light detectors. And in addition, we add a microwave antenna to excite our qubits, to control the spins, to excite coherent superpositions. And if you now measure the brightness of this spot in fluorescence intensity and sweep your microwave field, you will see a sharp response at the frequency of spin transition. The sharp response is indicating energy of the transition, and this is kind of a key tool now for turning your qubit into sensor. So this is a picture of experiments. This all can be very small and very integrated. The heart of experiment is here, and this area of diamond. Here is a piece of synthetic diamond with a microwave antenna on top of this. Color centers are implanted here, and we then are going to attach molecules to this area. So one word about sensing, how you can sense with qubits. The energy of the spin qubit depends on the magnetic field. So if you apply magnetic field, you will see the splitting of the spin transitions. You can do it on a single qubit level, and the precision of B-field measurements, the accuracy, the sensitivity, depends how sharp are these lines are. The longer is coherence time, the better you can measure the splitting, and the more precise your sensor is. So it can be very precise if you have a long coherence time, and so that's why basically coherence here is playing a key role to reach high enough sensitivity. Now, example of biosensing. I show here a collaboration between our group and a group of Michael Lukin at Harvard University, where the protein, a biomolecule, was attached to the surface of diamond, and in this center was interacting by magnetic dipole-dipole coupling with nuclear spins inside of this protein. In this case, it's not an induction signal that was detected by the coil, but rather the B field itself measured by NV center. So there are a few, of course, key elements. It's not only about having a good sensor, but also attaching it to the biomolecules. And this kind of classical biochemistry is also very, very essential. So here is an example of very clean diamond with NV centers sitting behind the surface. And then there was a special chemistry process applied, leading to attachment of proteins. It's close to monolayer, but the sensor itself is so close to the biomolecule that do see only the closest molecule, the molecule which is located inside of the sensing volume. And this sensing volume is just a few nanometers. Actually, technique itself is so selective that it don't see the whole protein, but rather the fragment of it. And we also need actually to turn magnetometer into spectrum analyzer. This can be done by applying right coherent control tools. For example, spin echo, by applying exciting coherence of the sensor and applying many pi pulses, allow you to refocus all the frequency component of external field except one particular frequency. And this is given by time interval between pulses. So it's somehow similar to optical spectrometer, where you have a grating and you diffract the light of certain wavelengths in one particular direction because of phase matching between a kind of incoming light. And in this case, you kind of have a phase matching between your AC magnetic fields and the train of the pulses. So it's similar to optical spectrometer, but in time domain now. And with this, you can reconstruct, or you can construct actually the sensitivity function and sweep it through the spectrum. And here the example of nuclear magnetic resonance spectra of individual molecule. It has two frequency components because protein was having a deuterium kind of added in its nature. It's not really labeling because it's chemically individual, the same protein, but it's just to make sure that we detect right signals and we don't have a noise from a hydrogen around. So it was labeled by rar deuterium and rar carbon-13. And in this case, you have a frequency component associated with deuterium and carbon-13. So those are noisy spectra, and you might ask yourself, so whether this quantum sensor is really better than conventional nuclear magnetic resonance. But remember about the sensitivity gap. So we are talking about jump from a millimolar, 10 to the 20 molecules sensitivity to a measurements of a single molecule. Of course, it's measurements in a very small volume. It's hard to compare sensitivities in terms of molar, but in terms of number of molecules, this is a very big improvement. Although spectra are noisy and they measure it for a very long time, in this case about 3 hours, so it's not good enough yet to measure the dynamics of these molecules. So we need to improve the sensitivity to be able to capture, the motion of fragments of these molecules. And another point to improve is the spectral resolution, because we would like to see interaction between atoms inside of complex molecules or interactions between nuclear spins. And for this, there are several kind of benchmarks that need to be achieved. Currently, we can distinguish between different atoms, chemically different species. For example, we can easily distinguish between hydrogen and carbon-13, as you have seen before. Those are having very different frequencies. But coming to a chemical shift or J-coupling, or so very tiny interaction inside of molecules, this is still difficult with this quantum sensor. There are some fundamental limits, like, for example, a back action. If I measure a molecule, I need to measure it kind of efficiently, but in my sensor do have some fundamental limit on the perturbation it induces on the molecule. And there are some more technical limits why we cannot yet achieve this very high resolution. And this is ongoing field of research, and I'm sure that in next years we will kind of touch a little bit the area of structure reconstruction on a single molecule level. Another point is about interfacing. This was kind of in vitro experiment. Everything was in a solution, then molecule was attached. But molecules in biology and in medicine, they are really working inside of cells. So the relevant processes are happening not on a glass, but inside of living organisms. So we need to interface with the living cells, our sensors. There are several ways to do this. One is actually grow the cells on a layer of NV centers containing diamond, and then you would try to basically see via long-range dipole-dipole interaction molecules inside of cells. It will not possibly work for very long distances, but can work in different settings from a few nanometers, where you can reach the lipids, to possibly a few tens of nanometers, touching a little bit area inside of cells. If you can see deeper and having less limitations, you need to bring the sensor inside of cells. And here, what is can be an interesting avenue is to use not a big piece of diamond, but rather nano diamond carrying these color centers. They can be taken by cells, And here I show you an image of the cell using fluorescence microscope. There are two colors here to see. The green color showing the shape of the cell is reflecting the fluorescence of dye, which was embedded in a cell membrane. But the red dots here are in V-center emission in nanodiamonds, and now this can be via functionalization attached to a group of interest and used for this type of sensing. Nuclear magnetic resonance can be one area of this sensing, but it's not all. There are wider spectrum of applications. If you have a sensor inside of living cells, you can think about measuring a current through the membrane channels. It's about a 10 picoampere field. For the current of 10 picoampere, you have 100 picotesla field associated with this current if you are very close. You can think about sensing biochemical reaction by measuring catalytic centers in enzymes, and they are carrying electron spins and can induce a microtesla fields. This is all within a sensitivity range. You can also think about sensing more complex processes, for example, propagation of action potential in a neuron network. Every time where you get an electrical signal passing through your neuron, there is also a current flowing through, and current is inducing a magnetic field. So capturing a magnetic field tells you also information about signaling in cells, and this process in many settings is not fully understood. So here you have a new tools. In addition to electrical detection, which is currently a golden standard, you have now a magnetic sensitivity window. So there's new signals, and this is very important. There are a few proposals on table how to do this neurosensing, but essentially it all comes to placing cells on a diamond chip. And so you can also now simulate in theory what could be the signals, and it seems to be within sensitivity range of the sensor, even on a single neuron level. So far, it was very difficult to do it on a single neuron level. So that's such that the magnetometer should be within sensitivity range. And recently, actually, first demonstration came up. I show here the recent work by a group of Ulrik Andersen and Alexander Hook at a Technical University of Denmark. So they took a slice of the brain, and so it's of course animal experiments. So it's more preclinical studies, and the slice of the brain, being still alive, coming from the mouse, was deposited on a diamond chip, and then magnetic field was measured upon stimulation together with electric field or electric signals, and both signals are corresponding. They are not one-to-one corresponding to each other because magnetic signal is richer, it has a longer range. Electric signals is only sensitive to a surface of the cell, and with magnetic detection you can see deeper inside of tissue, and this can provide a new way of studying the activities and signaling inside of neuron cells. And usually what is interesting is not really looking on a single neuron, but looking on communication between neurons, how these waves are propagating. That can have a fundamental importance in understanding how brain works. It also can have diagnostics importance in medicine, because the way how neurons are communicating can be related to diseases or success of treatment. In the last few minutes, if I have time, I think I will touch on other parameters that sensors can give you, and namely temperature. Temperature is a very essential parameter in biology and in medicine, and ability to measure temperature at nanoscale inside of cells. Of course, in the microscopic scale, it's trivial to measure it, but in nanoscale, it's something which is not very well developed. But there's few techniques available. Some of them are very precise and calibrated, like, for example, thermocouples or calorimeter, brought inside of cell, but this damages the cell. So you can bring your very sensitive classical temperature sensor inside of cell by damaging it. And so, but then you can measure temperature precisely, but you might basically lose ability of cells to function right. More recently, fluorescent dyes were proposed and demonstrated as nanoscale temperature sensors. Fluorescence in these dyes depends on temperature. And there were some very interesting results reported showing that inside of cell you can have a very different temperatures. However, there are some artifacts related to sensitivity of dye fluorescence, not only to temperature but also to other parameters, like for example a pH. And there was also discussion in the community whether this measurement that were reported are really free of artifacts. A thermodynamics model based on some very basic kind of boundary conditions tells that you don't expect more than a few millikelvin temperature gradient inside of cell. On the other hand, reported results were showing some degrees of temperature difference. And so there are also development of making a spin qubit to be temperature sensitive. Of course, spin itself is not really sensitive to a temperature, but narrow spin transition in diamond for a triplet state is sensitive to compression of diamond lattice. And diamond lattice expands with the heating. It's tiny effect, but long coherence time allow you to track tiny frequency changes. And so here I show you the change of this spectrum associated with the spin. If you heat your cells or biotissue by 10 degrees, so you can track this change and you can track it very precisely, much better than the line width. So current sensitivity of this technique can be on the order of 10 millikelvin. So this is one new tool. There is even simpler technique based on a shift of optical transition of color centers. So this is emission of one of the color centers in diamond, and so you can track the change for a few fraction of degree changes within a second of measurement time. And so this can be now translated inside of cells. Here's example again of our cell containing now nanodiamonds with this temperature sensitive optical lines. And you can track the small changes when the nanodiamonds propagate from one cell area to another. And so now this temperature change can be tracked in real time in a very calibrated way, because the spin transition and optical transition of color center inside of nanodiamond are very well shielded from other influences inside of cell. All right, I think with this I conclude. Just to highlight again the main avenues. So we were focused on nanoscale sensing of biological processes using spin qubits as a sensor. So this is some subfield. It's not all the biosensing, but it's quite interesting in terms of measuring molecules in very low concentration, recognizing its structure and dynamics. So it can have applications in diagnostics, also understanding of metabolic process. And so far, it's mostly demonstrations in the lab. And it's not yet at the level where it can be applied in a routine diagnostics procedures, like, for example, in a point of care and in a hospital. And so basically integration these technologies into compact devices can be very, very essential. Last but not least, I was focused on basically magnetometers for nanoscale sensing. Also, microscopic magnetometers can be very essential for biology and medicine, and detection of brain activity can be done also in a clinical settings, provided that you detect activity not on a cellular surface, not in a nanoscale, but outside of the brain. With this I conclude, and I think we have a few minutes for questions. Yes.
So you said that you are sensing classical objects, right? But your spatial resolution becomes so high
Yeah.
that maybe at some point, do you expect at some point there will be deviations of these thermodynamic parameters that is temperature, entropy from classical quantum thermodynamics to
Right. Yeah. So in a so the question was, because it's nanoscale sensing, they can be intrinsically quantum objects, sort of. So even a classical in a large scale, they can become quantum on a small scale. Indeed, I think it's indeed very, very interesting regime. Also in temperature, so in a thousand of atoms or thousand of molecules, fluctuations of temperature that are relevant. Also for the spin spectroscopy, actually, in many settings, the sensing is based on measuring fluctuations. So the signal itself is coming on fluctuation. So it's already touching this kind of non-equilibrium dynamics. Just that we were not sensing like some coherent processes inside of biomolecules. This is more difficult, or maybe was not the scope here. But having non-equilibrium dynamics, it's already within reach. Yeah.
When you do the temperature measurement, do these measurements themselves introduce some energy in the sample at the point where you want to measure?
Yeah, it's a very good point how non-invasive it is in terms of like heating. Yeah, laser might introduce some heating. So actually, there is one intrinsic heating process. You have to excite your color center with light. It falls down and leave a little bit of energy in a lattice. So this process is kind of negligible. So if you calculate the temperature rise associated with this process, so it's very small. So this is some fundamental but very small effect. So it will be on the order of nanokelvin or even lower.
Okay.
So because it's just one atom, there is not so much energy. But there are parasitic effects. As if you have some absorbing layers on diamond or inside of cells, and so you send the light, there is absorption of some unknown molecules. So that can lead to larger effect, and this is under investigation. It looks like, so this is something within a millikelvin, but it definitely exists, and it depends on the wavelengths of light also you are using. Yeah.
Okay, thanks.